Okay, so welcome to this next video in which we're discussing bradykinin-induced vasodilatation. So far what we've seen is that bradykinin is going to come and bind to this B2 receptor, and this is going to happen all over the surface of the endothelial cell. It's going to lead to the activation of the alpha-Q subunit, which means that the alpha-Q subunit now has GTP bound to it. Now, what is alpha-Q GTP going to do? Well, basically, it's going to activate an enzyme in the cell membrane. So let me draw a bit of the phospholipid bi there down here. Okay, so there is an enzyme implanted in the cell membrane known as phospholipase C, and it's specifically phospholipase C of the beta type. Okay, so this is the enzyme phospholipase C, and it's of the beta type. Okay, and it's basically going to be activated by the alpha-Q GTP subunit. So this is going to come over here, and it's going to activate this enzyme. So it's going to bind to it and activate it. Okay, so what color should we have phospholipase C beta drawn in? We'll have it in orange. Now, all phospholipase C enzymes break down phospholipids, and specifically what phospholipase C beta is going to do is it's going to break down an ordinary component of the phospholipid bilayer known as PIP2. So I now want to have a little bit of a discussion about what PIP2 is, because it's one of these molecules that people generally have no idea. So I want to, they've heard of it before, but they don't actually know what it is. So I want to talk a bit more about what it actually is. So, in order to do this, let's firstly remind ourselves of what a normal phospholipid structure is like. Now, we'll draw little cartoons for this. So the normal structure of phospholipid is that it looks kind of like this, okay, with a phosphate head here. So it has, let me explain what this diagram means. It has a glycerol molecule here, which is the backbone for the entire thing. So in green here, this is glycerol, or the chemist's name for glycerol, the proper name for glycerol, is that it's propane 1,2,3 triol. Okay, so propane 1,2,3. I suppose I should put that there, triol. Okay, which means that you have a free carbon molecule, and of every single carbon you have one hydroxyl group. Okay, now to the first two hydroxyl groups, what you have done is you have esterified them with long-chain fatty acids or long-chain carboxylic acids. So you've bound fatty acids or long-chain carboxylic acids to the hydroxyl groups of the first and second carbon of your glycerol molecule. And the proper name for fatty acids is that they are just long-chain carboxylic acids. Now, these make up what are known as the hydrophobic tails of your phospholipid molecule, long-chain carboxylic acids. And these hydrophobic tails, they all interact with each other in the hydrophobic core of the phospholipid bilayer, and they let these phosphate heads, as we'll see in a moment, stick out towards the cytoplasm and the extracellular fluid. Okay, so now we'll colour in that phosphate head over here. So this is a phosphate head which you have esterified in a phosphoester bond with the third hydroxyl group. So this is a phosphate head. Right, phosphate head. So these phosphate heads then have a negative charge, and they therefore interact favourably with water. So they are sticking out towards the cytoplasm or the extracellular fluid on either side of this phospholipid bilayer. Right, now, this structure that I've drawn here, this is a phospholipid. The entire thing is a phospholipid because you've got a lipid structure with a phosphate head here, so that's why it's called a phospholipid. Now, phospholipids also have an old name, an old biochemical name, which is phosphatidate. Okay, so these molecules, you will sometimes hear them referred to as phosphatidate molecules, Now, you might wonder, well, why is he telling us that? Well, knowing what phosphatidate means is actually going to be very helpful when we come to try and understand what PIP2 is. And let me try and explain that now. So, PIP2, this molecule that phospholipase C beta is going to break down, what does it stand for? This is its shorthand. This is what you will hear people call it because it's nicer than the long name. But the long name, the full name, is phosphatidyl. And that's what this first P is. And then the I is inositol. Okay, so let me highlight this in. So P here stands for phosphatidyl, and I've told you what phosphatidate is, so you might be able to guess that there's some relation between the two, and there is. Inositol we'll have in blue, that's the I there. And then it's not quite just phosphatidyl inositol, it's 4,5-bisphosphate. Bisphosphate. Okay? And 4,5-bisphosphate is then abbreviated to this P2. So you've just put the P2 there to indicate that you've got two phosphate groups sticking off it. So that's this 4,5-bisphosphate. Okay, so what is this molecule? What is it in terms of this? Well, basically, it's a modified version of this. So, we draw out the phosphatidate molecule again, okay? So here we have it. So, here are these two long-chain carboxylic acid tails, esterified to our glycerol molecule here, and with this phosphate head coming off at the front here. That's a phosphatidate molecule, or a phospholipid. What we want is phosphatidyl inositol. So we want this group stuck off an inositol molecule. And the way you do that is you stick the inositol molecule here. Or you could, the way it's named, it's as though you start with inositol and then you stick off phosphatidyl, the phosphatidate group. But really, the phosphatidate group is bigger, so it really should be inositol stuck off phosphatidate. Okay, so here's the inositol group. Now, inositol is a 6-membered carbon ring, so it's a hexagon structure. And every single member in here is carbon, basically. And all of the bonds between the carbons are single bonds. That means that all of the carbons need to have two groups coming off them. And they all have one hydroxyl group coming off. So every single carbon has a hydroxyl group coming off it. So you overall have 6 hydroxyl groups. And then every carbon also has a hydrogen coming off it. So it's a very nice rotationally symmetric molecule. Okay, so this is inositol here in blue. Okay, so this 6-membered carbon ring will associate like that. So now, what you have got there is phosphatidyl inositol, or PI for short. I'm sorry about managing to spit all over that. Right, and then, to finish this off, what you need to do is you need to add 2 phosphate groups onto it, and you need to add them on to the 4th and the 5th carbons of inositol, basically. So, here's the 4th carbon of inositol here, and the way you label it is you go 1, 2, 3, 4, 5, 6, and this is the 5th carbon. Now, for the life of me, I do not know why this is not called phosphatidyl inositol 3,4-bisphosphate, but for some reason, someone long ago decided that the carbon which has this other phosphate group here should be the 5th carbon rather than the 3rd carbon. I don't know why it's called that. We're stuck with it though. So, phosphatidyl inositol 4,5-bisphosphate. You've got this phosphate of the 4th and the 5th carbon. Right, so this is the usual constituent of the phospholipid binder, and basically I hope what I've shown you here is that all it is a normal phospholipid with an extra bit stuck off the head. So it will have an extra little bit stuck off, sticking into the cytoplasm basically. But apart from that, it's just a normal phospholipid. Okay, now, what is phospholipase C beta going to do to this PIP2 molecule? Well, basically it's going to break it down. So let me show you this. What it's going to do is it's going to cut this bond here, the bond between the glycerol molecule and the phosphate group, which was a phosphoester or a phosphate ester bond. So, what 2 molecules are you now going to get when you break that bond, when you hydrolyze that ester bond? Or phosphoester bond? Well, you're going to get a glycerol molecule with 2 fatty acids still esterified to it. So here it is. You've got the glycerol there, with 2 fatty acids still bound to it, but now it's got this free hydroxyl group on the 3rd carbon, okay, and this is what is known as diacyl glyceride, okay, diacyl glyceride, which is often just abbreviated to DAG. Now, the reason it's called diacyl glyceride is, well, the glyceride is for glycerol, obviously. Diacyl, the other name for carboxylic acid groups, which is what these fatty acids have, is an acyl group. Well, it's slightly more complicated than that, but once you've taken the hydroxyl group off a carboxylic acid group, so let me just show you this. So if we draw a carboxylic acid here, okay, this is the carboxylic acid group here. Okay, so this is a carboxylic acid here. And R, if we were talking about fatty acid, it would have to be very, very long chain, basically. Okay, now, this group here, let me colour it in rather than just drawing it in some hideous black pen. Okay, this portion, this is what is known as an acyl group. And when you bind a carboxylic acid to a hydroxyl group in an ester link, you take off this hydroxyl group. So effectively, all you're adding on is this acyl group. Okay, so that is why this is known as a diacyl glyceride, because you've added 2 acyl groups onto your glycerol molecule. Okay, then the other molecule you get, and that's really horrible, it's sort of seeping over. I'm going to get a new piece of paper after this. Right, so the other molecule you get is inositol 1,4,5 trisphosphate. You get this inositol molecule with 3 phosphate groups added onto it, like so. Now, the full name for this is inositol, clearly, because you've got the inositol ring, which is the center of this structure, basically. It's the star of this structure. Okay, so here's the inositol ring. And then you've got 3 phosphate groups coming off here. So inositol 1,4,5, and then tris phosphate. So the way you label what carbons the phosphate groups are going off is you label this one the first, then you count 2nd, 3rd, 4th, 5th, 6th. So you've got phosphate groups coming off the 1st, the 4th, and the 5th. And again, I do not know why they didn't label this one 1, this one 2, this one 3, this one 4. But they decided that this carbon with the phosphate group coming off it should be the 5th carbon in this ring. And we're stuck with the name. So inositol 1,4,5 tris phosphate. Now, for short, people will often just denote this I for inositol, and then P3 for 3 phosphate groups sticking off it. Okay, so let me just draw these in. Okay, right. So overall, what we have done so far is we've activated this B2 receptor all over the cell. It's caused the activation of the alpha-Q GTP subunit, which has then activated our phospholipase C-beta, which is now breaking down this normal component of the cell membrane, PIP2, phosphatidylinositol 4,5-bisphosphate, into diacyl glyceride and inositol 1,4,5 trisphosphate. Now, both of these go on to have signaling effects downstream. We are not going to be interested in the signaling of diacyl glyceride. Diacyl glyceride goes on to activate protein kinase C, and it's very terribly important elsewhere. But IP3 is the one we're going to be interested in, because that is going to cause an internal calcium signal. And we'll continue this discussion in the next video.